Good day. I am Janine Kayabyev from Bachelor of Secondary Education, Block 2. Now I will discuss to you about mitosis. Mitosis is a type of cell division in which one cell, or the mother cell, divides to produce two new cells, the daughters, that are genetically identical to self. The major purpose of mitosis is for growth and to replace worn-out cells. If not corrected in time, mistakes made during mitosis can result in changes in the DNA that can potentially lead to genetic disorders. Mitosis is divided into 5 phases. Interphase. The DNA in the cell is copied in preparation for cell division. This results in two identical full sets of chromosomes. Outside of the nucleus are two centrosomes, each containing a pair of centrioles. These structures are critical for the process of cell division. During interphase, microtubules extend from this centrosome. The prophase. The chromosomes condense into X-shaped structures that can be easily seen under a microscope. Each chromosome is composed of two sister chromatids containing identical genetic information. The chromosomes pair up so that both copies of chromosome 1 are together, both copies of chromosome 2 are together, and so on. At the end of prophase, the membrane around the nucleus in the cell devolves away, releasing the chromosomes. The mitotic spindle is consisting of the microtubules and other proteins extend across the cell between the centrioles as they move to opposite poles of the cell. The metaphase. Metaphase is the chromosome line up neatly end to end along the center or the equator of the cell. The centrioles are now at opposite poles of the cell with the mitotic spindle fibers extending from them. The mitotic spindle's fibers attach to the sister chromatids. The fourth is anaphase. The sister chromatids are then pulled apart by the mitotic spindle, which pulls one chromatid to one pole and the other chromatid to the opposite pole. Telophase. At each pole of the cell, a full set of chromosomes gather together. A membrane forms around each of the set chromosomes to create two new nuclei. The single cell then pinches in the middle to form two separate daughter cells, each containing a full set of chromosomes with nucleus. This process is known as cytokinesis. The meiosis. Meiosis is a process where a single cell divides twice to produce 4 cells containing half the original amount of genetic information. These cells are sex cells, sperm in males and eggs in females. During meiosis, one cell divides twice to form 4 daughter cells. These 4 daughter cells only have half the number of chromosomes. On the parent cell, they are haploid. Meiosis produces our sex cells or gametes, eggs in females and sperm in males. These are the process of mitosis and meiosis. The prophase. Prophase is the first phase of mitosis, the process that separates the duplicated genetic material carried in the nucleus of the parent cell into two identical daughter cells. During prophase, the complex of DNA and proteins contained in the nucleus known as chromatin condenses. The metaphase. Metaphase is a stage of mitosis in the eukaryotic cell cycle in which chromosomes are at their second most condensed and coiled stage. These chromosomes carrying genetic information align in the equator of the cell before being separated into each of the two daughter cells. The third one is anaphase. Anaphase is the fourth phase of mitosis, the process that separates the duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. Before anaphase begins, the replicated chromosomes called sister chromatids are aligned along the equator of the cell on the equatorial plane. The sister chromatids are pairs of identical copies of DNA joined at the point called centromere. The telophase. Telophase is the fifth and final phase of mitosis, the process that separates the duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. Telophase begins once the replicated paired chromosomes have been separated and pulled to opposite sides or poles of the cell.